really just objective 1. So all we will get through for this third exam is going to be to learn the general anatomy of skeletal muscle. So pretty much the structure, as I mentioned before. Now, skeletal muscle is unique, right? It's the only muscle that we have voluntary control over. The other two muscle groups, which are cardiac muscle, smooth muscle, we do not have voluntary control over. Skeletal muscle is also unique in that it is innervated by motor neurons. And we looked at that when we looked at the somatic nervous system. So a lot of what we're going to speak about as we talk about skeletal muscle physiology is really a continuance from the understanding of the neuromuscular junction. The events that took place at the neuromuscular junction, the receptors, which are nicotinic, the neurotransmitter, acetylcholine. And we're really going to just continue on looking at that sequence of events, what happens on the muscle side in order to bring about muscle contraction. So we're going to start that conversation off by looking at the structure of skeletal muscle. So skeletal muscles are suspended in our body. They are connected by 2, sometimes more bones, depending on how many heads that muscle has. And they do so via tendon. So the connection from the actual belly of the muscle, and I'm going to call the fleshy part of the muscle the belly or the bulk of the muscle. And then there is a tendinous attachment onto that bone. And so we've got the movable part of the muscle, which is the actual belly, and we've got the tendinous attachment, which is not moving. That is simply dragging the muscle or dragging the bone, right? So we've got the contractile part and the non-contractile part. If we were to look at muscle a little bit closer, we would see several different compartments that have their own connective tissue coverings. The first one is the epimysium. Now, the epimysium is what covers the entire muscle body or the entire muscle belly. And that pretty much divides one muscle from another or muscle from bone. That's how we can identify the biceps muscle, for example, as an individual body of muscle because of that unique separating connective tissue covering the epimysium. If we were to look inside the epimysium, we would see several different repeating units, which we call muscle fascicles. And they are covered by the perimysium. So the perimysium extends within the muscle body, and it's going to divide the muscle into several different bundles, which we call fascicles. Now, they're about 100 to 1,000, anywhere in between that range. There's lots of different sizes of muscles, but we've got anywhere from 100 to thousands of muscle cells located inside each muscle fascicle. And so the muscle fiber is really the single cellular unit of muscle. We can also consider the muscle fiber as the muscle cell. And that is wrapped in another connective tissue layer, which we call the endomysium, okay? So we've got the epimysium, which surrounds the entire muscle body. If we look inside of that muscle body, we can find several different fascicles. Each fascicle is wrapped in the perimysium. And if you took a single fascicle and looked within that, we would see several smaller repeating units. These are the actual muscle cells or muscle fibers, and they are wrapped or encased with endomysium around a single muscle fiber or a single muscle cell. Okay. Let's talk about some of the other structural components of a muscle fiber. We've got several different nuclei. So several different nuclei within a single muscle fiber. So they are multinucleated. If you look at the cell membrane or the plasma membrane of a muscle fiber, that is called a sarcolemma. That is separating one muscle fiber from the other. And then inside of that plasma membrane, inside of that sarcolemma, we've got the cytoplasm. In this case, we're going to call it the sarcoplasm. And that is pretty much the fluid that is bathing the organelles within that muscle fiber. We can also consider this to be the cytosol. So the sarcoplasm, in the case of muscle, is what we consider the cytoplasm or the cytosol to be. All right, so here is a closer look at the structure of muscle and then the individual subcellular and smaller repeating units that make up muscle. So as we can see, we've got the entire muscle body, the muscle fascicle, the muscle fiber. Now, if you were to look even closer within that muscle fiber, there are actually smaller subunits, smaller repeating subunits inside a single muscle cell. We call these myofibrils, okay? And down at the level of the myofibril, this is really the functional unit of muscle. This is where the muscle function is brought about, down at the level of the myofibril. Okay. So let's look at what happens down at the myofibril. Let's talk about the structure of the myofibril. So we've got this several myofibrils, right? This is a single myofibril here. This is a muscle fiber. And we could talk about the different subcellular components that are located inside a muscle fiber along with these various myofibrils. We've got the sarcoplasmic reticulum. So this is the same thing as the endoplasmic reticulum. This is the smooth ER, or the storage area for calcium. And we can see that here throughout the various muscle fibrils, myofibrils. Now, the storage of calcium is going to be pertinent, crucial to the contraction of skeletal muscle. So it's really important that we point out the storage area here, the sarcoplasmic reticulum. We've also got numerous mitochondria. Now, mitochondria is also vital to muscle contraction. Okay, muscle is very energetically demanding, and so it's going to need lots of mitochondria to supply the ATP that's necessary to drive the cellular machinery that brings about muscle contraction. We can also look at the transverse tubules that are called the T-tubules. So if you look at this apparatus here, throughout the muscle, and we take a closer look at this area, this region is called a triad. And we can see two lateral sacs. Now, the lateral sacs are actually a part of the sarcoplasmic reticulum in blue here. And again, this is the storage area for calcium. And then down the center of this triad, we've got this T-tubule, or this transverse tubule. And this is pretty much a cleft, or sort of a channel or a pathway that runs deep into the muscle. And the purpose of the T-tubule is to spread the electric current throughout the muscle that brings about a synchronous muscle contraction. Okay, if you look at a different view of this T-tubule here, we can see that this invagination that goes deep within the muscle, and the purpose of this invagination is so that we can have a synchronous muscle contraction, because that action potential, that current, spreads throughout the muscle, goes deep within the muscle very quickly, and it uses these channels or these invaginations that we call T-tubules in order to do so. And then the lateral sacs I mentioned a little bit earlier are the terminal cisternae. This is how the calcium gets from its storage area and comes in contact with the T-tubule. And that is a really important connection which happens here at the triad. Again, very important for bringing about muscle contraction. So far you can kind of make these two notes in your, points in your notes. The T-tubule is pertinent to muscle contraction. The lateral sacs, where we've got the storage of calcium, is also very pertinent to muscle contraction. And we're going to come back to those two structures later on when we talk about the process that helps to bring about muscle contraction. Okay, so now let's look a little bit more closely at the actual myofibril. So the triad was actually surrounding the myofibril. Let's look at the myofibrils themselves. So these are what give muscle and skeletal and cardiac muscle their striated appearance. So we're going to see that this same type of repeating unit, the myofibril, in skeletal muscle as well as in cardiac muscle. Although cardiac muscle, again, is not innervated voluntarily, we still have the striations that make it appear sort of elongated, give it that striated appearance. We do not see myofibrils in smooth muscle, hence the term smooth muscle, right? We don't have these striated repeating units or myofibrils in that type of muscle. The myofibrils are actually orderly arrangements of two smaller filaments, actin and myosin. Actin we call the thin filament. It's made of a smaller or a thinner protein, hence we call it the thin filament. Myosin is made of a thicker protein, and so we call it the thick filament, okay? And these are the repeating units that we see along each of our myofibrils. And so the striations that make up skeletal muscle are really these repeating thick and thin filaments that are running parallel to each other that form the sarcomere, right? So the sarcomere is this singular unit. We're going to speak a lot about the details of the structure here, but the unit that's bordered between this line, this Z-shaped line on this side, and this Z-shaped line on this side, this unit is what we call a sarcomere, okay? And those are the repeating units that make up a myofibril and that are really important to the functional properties of muscle. So if we look even closer here, having a real histological appreciation of muscle, right? This is a really histological view, really subcellular structure that we're looking at here. We can speak about what's going on at the level of a single sarcomere. And we can describe this based upon different staining patterns that are appreciated histologically. So when the earliest physiologists that studied muscle took a piece of muscle, they put it under the microscope, they saw these different staining patterns, right? Some darker staining that we can see here, some lighter staining that we can see here, and then some intermediate staining that we can see here, right? This kind of gray, sort of moderate area of staining. And so the earlier physiologists kind of labeled or described these different regions according to their different staining patterns. And so when we looked at muscle a little bit closer and actually studied the different proteins that were making up these staining units, this is where we actually saw the machinery of muscle that helps to shorten the muscle that brings about muscle contraction. So we've got the A band. Now the A band is the dark band that overlaps thin filaments and thick filaments, right? So here we can see this entire stretch is called the A band. And we can see that the blue lines representing thin filaments are being overlapped by the red lines, which are representing thick filaments. And so this is that moderately staining region, right? Not too light, not too dark. And that is because there's a certain degree of overlap that's taking place between these two proteins, actin and myosin, which is giving that A band, that kind of moderate staining appearance. Then we can speak about the H zone. Now the H zone is an area of thick filaments only, so there's no overlap. And we can see here these red areas are called thick filaments or myosin filaments, and there's no overlap of the blue filaments or actin, so we call this region the H zone. Running down the center of the H zone, there's a single line that connects each of these thick filaments. We call this the M line. And this is really what is connecting one thick filament to the other, this line that runs down the center of the sarcomere. We can also speak about the I band. So the I band is really the periphery of the sarcomere. So it's going to be the outer boundaries of the sarcomere here. And this is only thin filaments, so it's going to be a little bit lighter staining, right? The thin filaments are much smaller or thinner proteins, so they're going to have a lighter staining pattern than would the thicker filaments. So right along the blue here, we can see the I band. And if we look at this histological image here, we can see the I band here, right? Then we can look at the Z line. The Z line is the line that connects the thin filaments or the actin filaments. And if we look here, we can actually see the Z-like configuration of this connection. So from one thin filament to the next, this is this Z-like or staggered configuration that forms the Z line, and that is running down the center of the I band. And so we can describe a sarcomere, which is the functional unit of muscle. This is the area that is crucial, or the area at which the shortening of muscle is really brought about. We can describe that region as being from one Z line to the next Z line. So the area that is bordered one Z line to the other is what we call a single sarcomere. And as we can see, there are many repeating sarcomeres along this myofibril, okay? And if we looked at a cross section of this myofibril, we would see two different protein filaments here. In blue, the thinner filaments are going to be actin, and then the thicker filaments are going to be our myosin filaments.